Welcome to Unit 3, where we'll explore the various instruments and media used in microbiology. A compound microscope is a fascinating instrument because it uses two sets of lenses, an objective lens and an eyepiece lens, to create a highly magnified image of tiny objects. Think of it as a powerful tool that allows us to see things far smaller than what the naked eye can detect. It's mainly used to observe things like cells, bacteria, tissues, and microorganisms. Basically, the building blocks of life, as well as fine structural details that are simply invisible without magnification. Now, when we say compound, we're referring to the use of more than one lens system. This design allows us to achieve higher levels of magnification and also improves the resolution, meaning we get clearer, more detailed images. So, in essence, the compound microscope is essential in many scientific fields because it enables us to explore the microscopic world in great detail, helping us understand the complexities of life at a cellular and molecular level. Let's dive into the principle of the compound microscope. At its core, a compound microscope uses two or more lenses to magnify small objects, allowing us to see details that are invisible to the naked eye. The key idea here is that light passes through the specimen, which is usually thin enough for light to transmit through it. The objective lens then gathers this light and creates a magnified real image. This image is then further magnified by the eyepiece lens, which acts like a magnifying glass for the image. So, essentially, the combined action of these lenses enables us to see tiny structures with great detail. Understanding this principle is fundamental because it explains how we achieve high magnification and resolution, which are crucial for scientific observation and research. This concept ties back to the overall theme of microscopy, using optical tools to expand our view of the microscopic world. Now, let's focus on the role of the objective lens in the compound microscope. The objective lens is positioned very close to the specimen, which allows it to gather light directly from it. Because it has a short focal length, it can bend the light rays significantly. When light passes through the specimen and enters the objective lens, it does a couple of important things. First, it collects and refracts the incoming light rays, bending them to focus properly. Second, it forms a real, inverted, and magnified intermediate image of the specimen inside the microscope tube. This image is formed at a point just beyond the focal length of the objective lens. What makes the objective lens so powerful is its high numerical aperture and short focal length. Together, these features enable it to produce high levels of magnification and excellent resolution. In essence, the objective lens is primarily responsible for creating the image we see and for the resolution, that is, how clear and detailed that image appears. This is fundamental to the overall function of the microscope, as it allows us to observe tiny details that are otherwise invisible to the naked eye. Now, let's focus on the role of the eyepiece lens, also known as the ocular lens, in the compound microscope. It's located at the top of the microscope and functions as a simple magnifying lens. Essentially, it takes the real image formed by the objective lens and acts on it as if it were an object itself. The eyepiece then magnifies this intermediate image even further, which is key to achieving the high level of magnification we associate with microscopes. The final image that we see through the eyepiece is virtual, meaning it cannot be projected onto a screen, and it's inverted, so the image appears upside down compared to the actual specimen. Despite these inversions, the image is highly enlarged and comfortable for us to view with our eyes. Usually, this final image is formed at the near point of distinct vision, which is about 25 centimeters away, or at infinity for more relaxed viewing. It's important to note that while the eyepiece increases magnification, it doesn't actually improve the resolution. That is, the clarity or detail of the image. Instead, its main role is to make the image larger so we can examine it more easily. This is a crucial point because it highlights how magnification and resolution serve different purposes in microscopy, and both are essential for detailed observation. Let's take a closer look at the principle behind the compound microscope. Light rays from the illuminator first pass through the condenser and diaphragm. The condenser's role is to focus the light, while the diaphragm helps regulate the amount of light that reaches the specimen. Once the light is properly focused and regulated, it passes through the transparent specimen placed on the stage. The objective lens then refracts these light rays, which results in a real, inverted, and magnified image inside the body tube of the microscope. This image isn't the final view. Instead, it acts as the object for the eyepiece lens. The eyepiece further magnifies this image, creating a final virtual and enlarged image that we observe. This entire process illustrates how the compound microscope uses multiple lenses and controlled lighting to produce detailed, enlarged images of tiny specimens, integral to our understanding of microscopic structures. Let's take a closer look at the compound microscope. This device is essential in biology for observing tiny structures that are invisible to the naked eye. It consists of multiple lenses that work together to magnify the image of a specimen, allowing us to see details like cell structures and microorganisms. The key point here is that the compound microscope provides high magnification and resolution, which is crucial for detailed study. Understanding how it works helps us appreciate the advancements in scientific observation and how they've expanded our knowledge of life at the microscopic level. This microscope is a fundamental tool that connects to the overall theme of exploring the unseen world around us. Now, let's take a closer look at the optical components involved in a microscope, starting with the eyepiece, or ocular lens. This is the part through which you, the observer, actually view the specimen. Its main role is to further magnify the image that has already been formed by the objective lens, giving you a clearer and more detailed view. Typically, the eyepiece provides a magnification of about 10 times or 15 times, which enhances the overall magnification power of the microscope. The importance of the eyepiece can't be overstated. It not only increases the final magnification, making tiny details visible, but also ensures that viewing is comfortable and the image appears clear. When designing or using a compound microscope, understanding how the eyepiece works helps us appreciate how it contributes to the detailed and precise observations that are essential in scientific research and education. Now, let's take a closer look at the objective lenses. There are several types, each designed for different levels of magnification. The low-power objective typically provides 10 times magnification, which is great for getting an overview of the specimen. The high-power objective offers 40 times magnification, allowing us to see more detail. And then, we have the oil immersion objective, which is 100 times. This one requires a special oil to improve the image quality even further. The main function of these objective lenses is to serve as the primary magnifying components of the microscope. They gather light from the specimen and create a real, inverted, and magnified image that we can observe. This process is fundamental because it forms the basis for what we see under the microscope. Their importance can't be overstated. High-resolution images and detailed observations depend heavily on these lenses. For example, the oil immersion objective increases the numerical aperture, which essentially means it captures more light and enhances the resolving power. This allows us to distinguish finer details in the specimen. So, in the context of the compound microscope, these objective lenses are crucial components that work together with the other parts to give us clear, detailed images. They're essential for scientific accuracy and for advancing our understanding of microscopic structures. Now, let's take a look at the mechanical components of the microscope, which are essential for supporting, holding, and enabling movement and focusing of the various parts. First, we have the base. The base is really the foundation of the microscope. It supports the entire instrument and provides stability and balance, ensuring that everything stays steady during observation. Without a solid base, it would be difficult to get clear and accurate images. Next, there's the arm. The arm connects the base to the main body of the microscope, serving as a support for the optical components like the lenses and the body tube. It's also the part we use to carry the microscope safely, which is important because these instruments can be quite delicate. Together, the base and arm form the core mechanical structure that keeps everything in place, allowing us to focus and manipulate the microscope effectively. These components are vital for the overall functionality and precision of the microscope, helping us examine specimens clearly and accurately. Now, let's take a closer look at some of the key components of a compound microscope. First, the body tube or head. Its main function is to maintain the proper distance between the eyepiece and the objective lenses. This is crucial because if the distance isn't just right, the image won't be clear. It also helps align the optical components, ensuring that the image forms accurately and sharply. Essentially, it keeps everything in perfect order for us to see those tiny details clearly. Next up is the revolving nosepiece, or turret. This part holds multiple objective lenses, each with different magnifications. The good thing about the revolving nosepiece is that it allows us to switch between these lenses easily. So, whether we need a lower magnification to get an overview or a higher one to see fine details, we can do so quickly and smoothly, making our observations more efficient. Finally, let's look at the stage. The stage is the platform where we place the slide containing the specimen. It usually has a central aperture, or opening, which allows light to pass through from the light source below. This passage of light is essential because it illuminates the specimen, enabling us to see it clearly through the lenses. The stage is fundamental because it holds our slide steady and provides the proper positioning for viewing. Together, these components work harmoniously to produce clear, detailed images, which is at the heart of what makes a compound microscope such a powerful tool for observation. Now, let's take a closer look at some of the key components of a compound microscope. First up is the stage clips or mechanical stage. These hold the slide firmly in place, ensuring it doesn't move around while you're viewing. The mechanical stage specifically allows for precise movement of the slide in both the X and Y directions. This means you can easily position your specimen exactly where you want it, making your observations more accurate and efficient. Next, we have the coarse adjustment knob. Think of this as your initial focusing tool. When you're first trying to get a general view of your specimen, you use this knob to move the stage or the body tube quickly. It helps you bring the object into a rough focus so you can see it clearly enough to fine-tune. And that brings us to the fine adjustment knob. This is used for the final, precise focusing. Especially at higher magnifications, sharpness becomes critical, and this knob allows you to make those small, careful adjustments needed to see the details clearly. Together, these components, stage clips, mechanical stage, and the adjustment knobs, are essential for getting a clear, stable, and detailed view of your specimen. They work together to enhance your overall viewing experience, making the microscope a powerful tool for your observations. Now, let's take a closer look at the light source and mirror. The main function here is to provide illumination, which is essential for viewing specimens clearly. Modern microscopes typically have built-in LED or halogen lamps, making them quite straightforward to use. However, older microscopes relied on mirrors to reflect external light, which means you had to position a light source outside the microscope and direct the mirror to focus that light onto your specimen. This highlights how technology has evolved to make illumination more efficient and user-friendly. Moving on to the condenser, its role is crucial in ensuring your specimen is well lit. The condenser concentrates and focuses the light onto the specimen, which enhances both clarity and resolution. Without a proper condenser, the image can appear blurry or dim, so it's a vital component for obtaining clear, detailed observations. Finally, the iris diaphragm controls how much light enters the condenser. By adjusting this diaphragm, you can regulate the brightness, contrast, and even influence the resolution of your image. It's like fine-tuning the lighting in a photograph to get the perfect shot. When used correctly, the iris diaphragm helps you achieve optimal viewing conditions, making your observations more accurate and detailed. Together, these components, the light source, condenser, and iris diaphragm, work seamlessly to ensure you get the best possible image quality from your microscope, tying into the broader theme of understanding how each part contributes to effective microscopy. Now, let's talk about how total magnification works on a compound microscope. Essentially, Total magnification is the product of the magnification of the objective lens and the magnification of the eyepiece. For example, if you have a 40 times objective lens and a 10 times eyepiece, multiply those together and you get 400 times total magnification. This means you're viewing the specimen at 400 times its actual size. Understanding this helps us see how microscopes can significantly enlarge tiny objects, making details visible that are otherwise impossible to see with the naked eye. Remember, the overall magnification depends on both parts working together, which is key to getting the best possible view of your specimen. The compound microscope is truly one of the most widely used laboratory instruments across various fields. Its ability to magnify and resolve tiny structures makes it indispensable in biological, medical, clinical, industrial, and educational applications. When we think about biological research, this microscope allows us to examine the detailed structure and organization of cells, tissues, and microorganisms. It's essential for observing processes like cell division, identifying cellular organelles, and studying physiological activities in both living and preserved specimens. In microbiology laboratories, the compound microscope becomes even more critical. It enables us to observe bacteria, fungi, protozoa, and algae, helping us identify and classify these microorganisms. This is vital for understanding microbial morphology and is also crucial for diagnosing infectious diseases accurately. Moving into clinical diagnosis, the microscope plays a vital role in hospitals and diagnostic labs. It's used to examine blood smears, urine, sputum, stool, and tissue biopsies. By doing so, it helps us detect pathogens, blood disorders, parasitic infections, and abnormal cells. So overall, the compound microscope is not just a tool. It's a cornerstone in advancing our understanding and diagnosis across many scientific and medical fields. Now, let's look at how the compound microscope plays a vital role in histology and pathology. In histology, it's used to study the microscopic structure of tissues, helping us understand how tissues are organized and function. Meanwhile, in pathology, it's essential for identifying abnormalities, whether it's cancer cells or degenerative changes, allowing us to diagnose diseases more accurately. Moving on to education, the compound microscope is a fundamental teaching tool across schools, colleges, and universities. It provides students with hands-on experience, helping them grasp complex concepts like cell structure, tissue organization, and microbial diversity through direct observation. This direct visual engagement makes learning much more effective. In biotechnology and molecular biology labs, the microscope is indispensable. It allows scientists to study cell cultures, monitor cell growth, observe genetically modified cells, and evaluate experimental outcomes. This detailed visualization is crucial for advancing research and development in these fields. Finally, in the pharmaceutical industry, the compound microscope supports quality control and safety. It's used to test for microbial contamination, analyze drug formulations, and examine drug crystals and potential contaminants. Ensuring the purity and quality of pharmaceutical products is essential, and the microscope helps make that possible. Overall, the compound microscope's versatility really underscores its importance across multiple disciplines, connecting the dots between research, education, and industry. Let's explore how compound microscopes are used across different fields. In forensic science, these microscopes are crucial for analyzing various evidence like hair samples, fibers, blood stains, body fluids, and even microbial traces. By examining these small details, investigators can piece together crime scene information and identify evidence more accurately. Moving on to environmental studies, these microscopes help us study water samples, soil microorganisms, planktons, and algae. This detailed examination is essential for assessing pollution levels and understanding ecosystem health. Lastly, in the food and dairy industry, compound microscopes play a vital role in ensuring safety and quality. They help detect microbial contamination, adulteration, and spoilage organisms, which is key to maintaining food standards. Overall, these applications highlight how versatile and important compound microscopes are in various professional fields, supporting critical analysis and quality control.